This is part C, the second part, under Unit 3, Cells. We'll look at the concentration of solutions. We'll discuss concentration gradients, and we'll compare passive and active transport processes. Let's look at concentration of solutions. Concentration is not the same as quantity or volume. Volume means the total amount of a solution, whereas concentration refers to the percentage of the parts of a solution. Usually we're dealing with solute and water. Concentration is measured in various ways: by weight, volume, molarity, percentage, and others. We have terms when we can use to compare concentrations of two solutions. Tonic, for example, refers to the solute concentration. Now, that's not actually what it refers to. It refers to the tension or pressure. You'll understand more about this a little later. Iso means same, hyper greater than, and hypo less than. Let's compare concentration of solutions. In a solution, if the term concentration is used alone, it refers to the solute concentration, as concentrated urine or concentrated fluid. If I use the phrase water concentration, it refers to the concentration or percentage of water. Let's compare three beakers, beaker A, B, and C. Which beaker has the greatest volume? Beaker C, 4,000 milliliters. Which beaker has the greatest solute quantity? Again, beaker C, 40 grams. Which beaker has the greatest solute concentration? This would be beaker A. Let's measure concentration in grams per 1,000 milliliters. What is the concentration in grams per 1,000 milliliters of beaker A? 20 per 1,000 grams, milliliters. Beaker B, 10 per 1,000. And beaker C, 10 per 1,000. Let's compare two solutions using three different beakers. The numbers represent percentages of solute in the three beakers. Let's combine iso, hyper, or hypo with tonic and put the answers in the blanks. Beaker number 2 is what compared to beaker number 1? Isotonic. Iso same, tonic solute concentration. What about beaker number 1 compared to beaker number 3? Hypotonic. Lower solute in number 1 compared to number 3. What about comparing beaker number 3 to beaker number 1? Hypertonic. Beaker number 3 has a greater solute concentration. What's the water concentration in beaker 1? 95%. After all, if the solution is composed of water and solute makes up 5%, the rest of it is water, the 95%. What's the water concentration of beaker number 3? 90%. What can you conclude about the water concentration in beaker number 1 compared to beaker number 3? The water concentration is greater than beaker number 1. You'll see why I'm emphasizing water concentration shortly. Concentration gradient means a difference in concentration. Think of gradient as meaning gradual. If you have a solution with a high concentration at one end and a low concentration at the other end, and you move from high to low concentration, you're moving down or with the concentration gradient. If, on the other hand, you move in the opposite direction, you're moving up or against the concentration gradient. Now remember, Particles are in constant motion and moving randomly in all directions, some up and some down, some toward you and some away from you. This motion is called kinetic energy. Now, it's different from metabolic energy. An electrical gradient also exists. This is the difference in charges and will be discussed in the nervous system unit. An electrochemical gradient is a combination of the two gradients. Let's look at the consequences of a gradient using a simplified model. We want to see what happens to movement if there's a difference in concentration. In our model, let's assume there are 4,000 particles on the left side and 2,000 on the right. Let's further assume that at this moment they're moving equally in only 4 directions instead of randomly in all directions. How many at this moment will be moving from the 4,000 side in each of the 4 directions? 1,000. At the same moment, how many will be moving from the 2,000 side in each of the 4 directions? 500. How many particles are moving from the 4,000 side, the left side, toward the middle? 1,000. How many particles are moving from the 2,000 side toward the middle? 500. What is the difference among those moving toward the middle? Now, this difference is the net. 500. 500 from 1,000 is 500. In which direction is the net movement? It's to the right. If a difference in concentration exists, the net movement of particles is from their high to low concentration. If you increase the difference in concentration, what would happen to the net movement? It would increase. Using the same model as before, let's double the concentration on the left side and see what the net movement in the direction is. The net movement would be 1,500 to the right. The greater the difference in concentration, the greater the net movement. Let's look at passive transport processes. In our model, we'll use a U-shaped container. Now remember, passive processes do not require metabolic energy. Take diffusion. In the simplest form, diffusion is the movement of particles. There is a net movement from high to low concentration. I don't like the definition many use for diffusion, which is the movement of particles from high to low concentration until the concentration difference is equal. That implies that diffusion stops when there's no difference in concentration. Diffusion is the movement of particles. It may be through a membrane. Dialysis is the diffusion of solute through a membrane. The middle figure shows dialysis. The membrane is selectively permeable because it allows solute to cross, but doesn't allow the water to cross. Osmosis, on the other hand, is the diffusion of water through a membrane. The membrane, again, is selectively permeable, but this membrane only allows water, but not solute, to cross. Facilitated diffusion requires a transport protein. Think of this as dialysis using a transport protein. It's a common form of transport of water-soluble particles because they can't cross the lipid bilayer. Glucose transport, in many cases, is an example. Filtration is the forcing of water and solute through a membrane by pressure. Let's look at osmosis, that is, diffusion of water through a membrane, in a little more detail. The water may pass through the lipid bilayer or through aquaporins. Remember, the solution is a mixture of water and solute. The U-shaped container has a selectively permeable membrane that allows water to cross. In container A, the solution on both sides of the membrane have the same water concentrations, and the diffusion of water through the membrane in both directions is the same. That is, there is no net movement. In container B, on the other hand, the water concentration on each side is different. 4,000 molecules of water, let's say, are on the left side and 2,000 on the right side. But to the naked eye, the water level in both sides of the container are the same. How can this be? When you add 2,000 molecules of a solute to the right side, it takes up space otherwise occupied by the water. Now the right side contains how many total molecules? 4,000. Notice that the solute has one function, and that is to take up space otherwise occupied by water. Solute does not push or pull water through the membrane. Since the water concentration in container B is different on both sides, what can you conclude about the net diffusion of water through the membrane? 500 to the right. Note that in both containers, water is moving toward the right and toward the left by osmosis, but in the container B, more water is moving toward the right. The difference is the net. What will happen to the solution levels on the left and right side of container B? The left side will drop and it will rise on the right side. If this is container B, the net movement of water is to the right. The solution level on the left side will drop and on the right side will rise. If you put a lid on the right side, what would happen to the pressure on the right side? Pressure would build up. This pressure is osmotic pressure, OP. What is osmotic pressure? Osmotic pressure is a potential pressure due to osmosis. Osmotic pressure does not cause the movement of water through the membrane, but results from the movement. Osmotic concentration refers to the solute concentration. Osmotic concentration depends on the number of solute particles, but not the size of the particles. Osmotic pressure is directly proportional to osmotic concentration. If the osmotic concentration increases, what happens to the osmotic pressure? It also increases. Let's look at the relationship between osmotic pressure in a solution and a cell. Here are three beakers having different concentrations, which is represented by a fraction, the water concentration, for example, 95, over the solute concentration, for example, 5. In each beaker is a cell. The cells have a concentration of 95% water and 5% solute. Now remember, cells are surrounded by a selectively permeable membrane. Water easily diffuses into and out of the cell by osmosis. What do we call the beaker relative to the cell if the beaker is 95% water and 5% solute? The solution in the beaker is isotonic. What if the beaker is 90% water and 10% solute? The beaker in the solution is hypertonic. If the beaker is 98% water and 2% solute, the solution in the beaker is hypotonic. In which direction is the net movement of water in beakers 1, 2, and 3? In beaker 1, there is no net movement because the concentration of water inside and outside the cell is the same. In beaker 2, The direction of movement is out of the cell from high water concentration to low water concentration. And in beaker 3, the net movement is into the cell from high water to low water concentration. What will happen to the cells in beaker 1, 2, and 3? In beaker 1, nothing. In beaker 2, the cell will shrink and die. In beaker 3, the cell will swell and burst. Let's look at the characteristics of osmotic pressure. Osmotic pressure is not the same as a physical pressure. For example, a tire may have pressure of 35 pounds per square inch. That means the air in the tire is pushing against the wall of the tire at 35 pounds for every square inch of surface. Atmospheric pressure at sea level is 14.7 pounds per square inch. So every square inch of your body has 14.7 pounds of air pressure. Of course, you're pushing back at the same pressure, so you don't notice the difference. Osmotic pressure of a 1 molar solution is 22.4 atmospheres. That's something like 329 pounds per square inch. The osmotic pressure is determined by the number of particles. As we said, not by the size of the particles. It's measured in osmoles. The osmole is the number of particles in 1 mole of solute if it does not dissociate or ionize when added to water. Since cells are so small, we use 1/1000th of an osmole, which is a milliosmole. If a solute dissociates, the number of osmoles would be multiplied by the number of dissociated particles. Osmolarity and milliosmolarity refers to the osmoles per liter. In living systems, the osmotic pressure is about 300 milliosmoles per liter. In solutions that are used clinically, again, living systems have an osmotic pressure of 300 milliosmoles per liter. A 5% glucose solution, called 5D5W, is an isotonic solution to living fluids. 1 mole is 180 grams of glucose. 1 osmole would be how many grams? 180. 300 milliosmoles would be a 5% solution. 0.9% saline solution, called normal saline, is also isotonic to body fluids. 1 mole of salt, sodium chloride, is 58 grams. 1 osmole would be how many grams? Half of that, 29 grams, because sodium chloride dissociates into sodium and chloride ions when added to water. 300 milliosmoles would be a 0.9% sodium chloride solution. Active transport processes require metabolic energy, and they include active transport itself. It requires energy from ATP. In fact, something like 40% of the energy that a cell produces is used for active transport. It transports against a concentration gradient. The most common example is the sodium-potassium pump. I often simply refer to it as the sodium pump. Many other pumps exist for other ions, for amino acids, for glucose, and so forth. Primary active transport pumps a substance directly through the membrane. Secondary active transport often uses a sodium pump to cause the second substance to cross the membrane. Symport, second substance, crosses in the same direction as sodium, and antiport, the second substance, crosses in the opposite direction as sodium. In exocytosis, this is a form of vesicular transport. We'll see why in a moment. And endocytosis is another form of vesicular transport. These are both active transport processes that require energy. Endocytosis includes phagocytosis, pinocytosis, and receptor-mediated endocytosis. Let's look at active transport. The general mechanism goes something like this. The carrier will bind to the ligand, that's the molecule being transported. The carrier is phosphorylated. The carrier's shape is or position is altered. In this case, it flips to the other side of the membrane, and the carrier releases the ligand to the other side of the membrane. What about the sodium pump, properly called the sodium-potassium pump? 3 sodiums are pumped out in exchange for 2 potassiums to move in. The sodium pump is critically important. It regulates cell volume. It's involved in secondary active transport. It produces heat, and it's necessary for membrane potentials, and we'll cover this in some detail when we get to the nervous system. Endocytosis and exocytosis, both forms of vesicular transport, are special processes that transport very large molecules like proteins or polysaccharides and entire cells and cell fragments. Let's look at exocytosis. This is how cells secrete materials. The cell is shown below, and part of it is enlarged on the right. Ribosomes make the proteins. The rough ER makes the lipids and processes the proteins because the proteins from the ribosomes have been inserted into the rough ER. Transport vesicles transport the molecules from the ER to the Golgi apparatus. In the Golgi apparatus, the molecules are processed, sorted, and packaged. Secretory vesicles, shown in blue, transport molecules from the Golgi apparatus to the plasma membrane. Often calcium is required for this process. The secretory vesicles may also transport molecules to the lysosomes. Some vesicles fuse with the plasma membrane and release their contents, whereas other vesicles pop out of the cell whole. This is commonly found in glands. Endocrine glands secrete substances into the blood. Exocrine glands secrete substances to a surface through duct. Let's look at endocytosis. This is how cells take in material. Will enlarge the lower part of the cell. General characteristics include adhesion. The particle to be ingested sticks to a receptor on the surface of the cell. Then the particle is ingested. The plasma membrane of the cell actually sinks inward and then encloses the particle, and it's called a food vacuole or a phagosome. What are these particles? Well, they might be microorganisms that a phagocytic cell is trying to remove from the body. It might be a fragment of cells, and so the cells are trying to clean up the mess. Phagosome will then fuse with a lysosome, and the lysosome will digest the particle. Why does that happen? Well, the lysosome is filled with very potent substances like hydrolyzing enzymes, free radicals, nitrous oxide, and many others. When the lysosome fuses with the phagosome, the potent enzymes then come in contact with the contents of the food vacuole, and the contents are digested. When they're digested, then they're transported through the phagosome membrane into the cytoplasm. Any undigested fragments then are released from the cell by exocytosis. You might wonder, why are the hydrolyzing enzymes and other substances located in the lysosome rather than simply free in the cytoplasm? Well, the reason is if they were free in the cytoplasm, they would digest the cell. Now, some cells, especially those in the embryo, naturally die as part of growth of the organism. They do so by the lysosome rupturing, releasing the contents, and digesting the cell. What about phagocytosis? Phagocytosis ingests large particles and cells into the phagosomes. Usually occurs in phagocytes that include macrophages and neutrophils. Pinocytosis, a form of bulk phase endocytosis, ingests molecules and fluids. It occurs in all kinds of cells. It does not require receptors. Receptor-mediated endocytosis does require receptors, and it ingests specific molecules. It's very selective and does not ingest amount of fluid. It's more complex than other forms of endocytosis. And this is the end of the second part of Part C, membrane systems. You're now ready to progress to the third part.